thank you. Thank you for that nice introduction and for giving me the chance to come and present some of our work. And also, I'd like to thank the people from the University of Essex, of course, for their great presentations today. So I would like to talk to you today a little bit about what we've been working on, a disease called type 2 citrullinemia, or often called just citrin deficiency. There's really two age-dependent phenotypes of this disorder. One occurs when children are just born, and it's classified as the neonatal intrahepatic cholestasis caused by the citrin deficiency. And this disorder typically is exhibited by a prolonged jaundice of the children, but also they have hypoglycemia and fatty liver. Typically, this disorder sort of clarifies itself in the first year of age, and these people enter what's called the apparently healthy period. However, these symptoms persist, such as fatigue, hypoglycemia, or even epileptic seizures. Later on, usually when the people mature, they develop the full-blown type 2 citrullinemia, and these symptoms include basically overaccumulation of ammonia, also abnormal behavior such as confusion, aggression, hyperactivity. Often this disorder can result in seizures and coma. So it's really a prolonged chronic disease. We also think that these patients who are in the apparently healthy stage probably are suffering from sort of the chronic damage that will ultimately manifest itself as the full-blown type 2 citrullinemia disease. So really the clinical features of type 2 citrullinemia is really the sudden onset liver failure. Often the diagnosis of this disease only occurs when people are really in the late stage of the disorder. These patients can often die from overaccumulation of ammonia, which can often cause swelling of the brain. Sometimes these symptoms are provoked after certain medications, infection, alcohol consumption, but especially after ingestion of a high carbohydrate diet. This seems to be a very important step in the disease progression, is the uptake of a large amount of carbohydrates. The biochemical features really result in decreased aspartate, increased ammonia, and interestingly, a disruption in the NADH-NAD ratio. So these patients have elevated levels of NADH and lower levels of NAD+, and this was mentioned earlier that the lack of the NAD+ can lead to a lot of defects in energy production and other metabolic pathways. So typically there's several dietary guidelines for these patients. But however, these dietary guidelines are very different from other patients who have these sort of urea cycle disorders or hyperammonia. So typically people with with excess ammonia are restricted to a high carbohydrate, high fat diet, low in protein, because the protein is often the source of the ammonia. However, people with the type 2 citrullinemia disorder, they cannot have a high carbohydrate diet because this will actually cause the disorder to become even more severe. So they're limited to a low carbohydrate diet that's rich in proteins and fat. So this becomes very important when these patients exhibit symptoms. Often the clinical features of the elevated levels of ammonia will often cause clinicians to immediately suggest this type of a diet, a low protein diet, high carbohydrate, high fat diet. However, this is like very bad for the patients. If the clinicians give this type of a diet regimen, this will lead to an exacerbation of the disorder, and has been shown to lead to death in many patients. So many patients in Japan who were given this sort of diet actually died from excess ammonia. So it's very important to have the right type of dietary intervention. So the diagnosis and treatment of the citrin deficiency are not especially good. You can get an initial diagnosis based on ammonia levels in the blood, or also changes in the citrulline or arginine levels. However, a definitive diagnosis requires sequencing of the gene that's involved in citrin deficiencies, SLC25A13. And this gene encodes the citrin, also called AGC2 or aspartate glutamate exchanger 2, and this is the only gene whose mutation is known to cause the citrin deficiency. Currently, the treatments are somewhat limited. The dietary intervention can limit the defects associated with this disease. In severe cases, liver transplantation is the only effect. Currently, addition of sodium pyruvate, intravenous sodium pyruvate, has been started. However, the efficacy of this is still not known. So the treatments and diagnosis of citrin deficiency are not well developed. However, this disease is apparently quite common, especially in East Asia. So the carrier frequency of citrin mutations in that SLC25A13 gene, are actually quite common. And several studies have shown that in Japan, the carrier frequency is about 1 in 60. In China, it's about the same. Korea and Thailand have about the same, about 1 in 100. So this is the carrier frequency, not the disease frequency. However, the disease frequency is estimated to be about 1 in 100,000 individuals in East Asian populations. So there's a profound discrepancy in the carrier frequency versus the disease frequency. We think this is actually due to a secondary mutation in another genetic locus that's probably required for the full-blown citrin deficiency disease. But the carrier frequency is quite high. Even though these mutations occur very, very often in East Asian populations, there are examples of people in Middle East, Europe, or even North America who contain mutations in the citrin gene. So even though it is quite common in East Asia, it does occur in other populations, non-Asian populations in these locations. So as I mentioned, the citrin gene, the SLC25A13, is a mitochondrial aspartate glutamate exchanger. So it's present on the mitochondrial inner membrane, and it exchanges aspartate for glutamates. So it transports aspartate out of the mitochondria and glutamate back into the mitochondria. It has two major biochemical functions. One is in the urea cycle, and the other is in what's called the malate aspartate shuttle, and I'll describe these in a little bit of detail. So for the urea cycle, the major function of citrin is to export aspartate out of the mitochondria. Aspartate can then combine with citrulline, ultimately produce arginine, which can be decomposed into urea, which can be excreted. In the absence of the citrin gene, or AGC2, the aspartate levels are decreased, cannot combine with citrulline, and now the ammonia cannot be detoxified through the urea cycle. So this is one of the defects associated with the loss or mutations in citrin, or AGC2. The other function of the citrin is the malate aspartate shuttle, and it performs the same function in that it transports glutamate in and aspartate out of the mitochondria. But here it's involved in the movement of reducing equivalents into the mitochondrial matrix using malate. So NADH can be oxidized through the conversion of oxaloacetate to malate. The malate can be transported into the mitochondrial matrix, where it can now be reconverted to oxaloacetate with the reduction of mitochondrial NADH. However, in the absence of the aspartate glutamate exchanger, this cycle stops, and NADH tends to accumulate within the cytosol of these cells. And this overaccumulation of NADH has the effect of blocking many enzymes involved in glycolysis, and this is thought to be the cause of the carbohydrate toxicity. When patients have a diet high in carbohydrates, this will cause enhanced flux through the glycolysis, causing an overaccumulation of the NADH, and now these reducing equivalents cannot be transported into the mitochondria. And this seems to be one of the major defects associated with the patients who have the citrin deficiency disease. So our collaborators, Ajarn Dwangwadi at the Faculty of Medicine, Department of Pediatrics, she was doing some genetic testing with Thai infants that had the intrahepatic cholestasis from unknown causes. And she found that several patients had missense mutations in the SLC25A13 gene. One of the mutations she found was a mutation of the star codon, and then there were two other mutations inside the AGC2 protein, a proline to a leucine and an arginine to a glutamine. However, the effect of these mutations is not clear, because these mutations were brand new and had never been characterized previously. So we got involved in the project at this point to try to understand if these mutations were involved in the citrin deficiency disease, or if they were simply polymorphisms that didn't have any effect on the citrin function. So in my lab, we use the Baker's yeast, Saccharomyces cerevisiae, to try to study both defects, or to study defects associated with human disease, as well as look at mitochondrial function. So this was a good match for our lab. And one reason that Baker's yeast is a good model system for this type of work is that it's a very simple eukaryotic cell, has a high degree of conservation in many of the basic cellular processes, and very critically, many human genes associated with disease have a close homologue in the yeast cell. And again, one of the main features is they're very easy to genetically manipulate. So we thought we could use the yeast model system to try to test these novel mutations to see if they had any role in the citrin deficiency disease. And of course, there is a homologue in S. cerevisiae of the citrin, which is called AGC1. And to try to mimic the disease state in the Baker's yeast, we found that if we grew the yeast with acetate as the sole carbon source, this caused sort of a mimicking of the carbohydrate toxicity that was observed in the patients. It drove the formation of malate. Now, this malate needed to be transported into the mitochondria to be used in the malate aspartate shuttle. However, in the absence of the citrin or the yeast AGC1, the malate aspartate shuttle was blocked. The malate could not be transported into the mitochondria, and again, NADH sought to accumulate in the cytosol. So we could use yeast grown on acetate as a model for the citrin deficiency disorder. What we found is that when we took the yeast lacking the AGC1 gene and expressed the human AGC1, we could complement the poor growth on medium containing acetate. So the blue bars are Baker's yeast expressing the human AGC, should be AGC2, sorry, expressing the human citrin, or the white bars of the gene deletion. And growth on acetate is fully complemented by the human gene back to the levels seen in the wild-type cells. However, growth on other carbon sources such as galactose, glycerol, or ethanol is not affected in the AGC1 deletion strains. It's clearly showing that it's not a general defect in mitochondrial function. It seems to be a specific defect associated with the growth on acetate, and we think due to the overaccumulation of the NADH. So we think that the yeast model certainly can function as to tell us about the function of the human AGC1 that's expressed in the yeast cells. So again, we can show that compared to the wild-type cells, these are just normal Baker's yeast expressing their normal copy of AGC1. Now we express the human citrin, or AGC2, in the AGC null strain, and we can see we get full complementation, fully restores the growth of these AGC1 nulls. So again, this gives us good confidence that we can use the model, the yeast model, to study the citrin deficiency disease. But before we could really say that we can study those new mutations, we wanted to validate our model system. And here we chose to study, actually I think I skipped some slides. We chose to analyze some previously characterized mutations in the SLC25A13 gene. So we looked at mutations that had been known to be associated with the disease but not been tested, or mutations that were found but not reported to be associated with the disease. So we mutated a residue here, proline 632 to leucine. Now this mutation was not associated with citrin deficiency but had been found in several patients. We found that that mutant was fully functional in our system, but mutations of other residues that had been implicated in the citrin deficiency disease showed very either reduced or entirely absent activity in our growth complementation assays. Now this was not due to changes in the protein levels. It seemed to be entirely due to changes in the protein activity. Also, we looked at localization in the mitochondria, and these mutations all seem to be localized correctly. So it seemed to be just activity, not protein localization or protein stability. So now we tested two of these mutations, the P502L or the R605Q, which were identified in the Thai patients. However, what we found is both of these mutations were completely active. They were not associated with reduced activity of the citrin. Again, the protein levels were the same, even though this proline at 502L is entirely conserved in every species we looked at. Mutation of this residue to leucine had no effect on activity. So we were actually quite surprised by this, but our results do clearly show that these two mutants, these two variations, are not necessarily associated with the citrin deficiency disease. So we were able to show that those two patients that were found that had the intrahepatic cholestasis were not suffering from the citrin deficiency disorder based on this analysis. We had one other mutation, was the PMET1, which is the loss of the start codon. So of course you would expect loss of the start codon to have a profound effect on the ability of this variant to function, and that is what we saw, was that loss of the start codon had completely abolished the ability of this variant to complement. So we can say definitively that this variation was a pathogenic mutation and does cause loss of activity of the citrin protein. One question we had, though, is that there was a possible alternative start site that would result in a truncated form of the citrin, a delta 1 to 34. We thought, well, perhaps in the patient's expression of this truncated protein may partially compensate. So we expressed just this truncated form, essentially expressed it to levels similar to that seen for the wild type, yet it still was unable to complement. And our work later we found is that loss of the first 34 residues caused a mislocalization of the AGC2 protein, mislocalization of the citrin protein. I just would point out at this point, while you may think this is an obvious result because of loss of the N-terminus, the mitochondrial carrier proteins do not exhibit the typical N-terminal targeting signals. Their targeting signals are in the internal structure, so loss of the N-terminus actually does not necessarily cause mislocalization. Loss of the complete N-terminus, about 200 residues, actually does not alter its localization, only loss of this first 34. But we found there was mislocalization of this delta 134 citrin that caused loss of complementation. So we thought, well, either way, either loss of the initial start codon, even if you were able to express this sort of truncated form, you would still have loss of function mutation. So we could really confirm that the PMET1 mutation would be a pathogenic variant. As I mentioned, further investigation of the patients that we had found with a P502L indicated that this patient had a disease called progressive liver fibrosis, not type 2 citrullinemia. The patient that had the R605Q variant, the disease that this child has not been identified, though I think we can rule out with our analysis that it was not the citrin deficiency disorder. Just one thing, looking back at the 502L variant that was quite active, we did some molecular modeling to try to see could we find a correlation between variants that were active and variants that were inactive. What we can see is that all the variants that we tested that were inactive are just shown in the red molecules. And we can see that most of them either occurred within the carrier cavity, or interestingly, some of them occurred on the very top and the very last helix of the transmembrane region. The effect of the mutants in the carrier cavity, I think, is obvious enough that they're probably interfering with either binding of aspartate or glutamate. The mutations at the very end are actually sort of puzzling, as they do not seem to be involved directly in substrate binding. The P502L here, we think the reason it has no effect is it's actually predicted to be present on the outside of the transmembrane region, not within the carrier cavity. So I think this sort of indicates that mutations that would be predicted to be within the carrier cavity certainly should have a profound effect on activity, but also mutations outside the carrier cavity, especially in this final helix, seem to be involved. So one thing that we found, as I was alluding to, is that even mutations in this sort of N-terminal loop, this extended N-terminal loop, are having an effect on the activity. Our PMET1 mutant was of course inactive, but in the course of our analysis of the PMET1, we found several other mutations that were involved in the calcium binding EF hand that altered activity, and also mutations that were really at the end this final transmembrane region seem to have some effect on activity, though mutations further on, the P632L or the R605Q, were not. So we actually are sort of interested in understanding more about these two regions that are not within the sort of substrate binding region to find out what their role is in the activity of the aspartate glutamate carrier. So as I mentioned, that PMET1 variant, it was actually the first time that variant had ever been identified. So we were curious to see, well, was it a common variant or is it uncommon? So again, our collaborator John Dornredy in the Department of Pediatrics, she found many, she did a screen where she sequenced several known mutations in the Citrin gene as well as the PMET1. And this was through several regions in Thailand, both the north, northeast, central, south, or within Bangkok. And overall, you can just look at the bottom line for the total mutants. She found that the previously characterized mutations had carrier frequencies of about 1 in 192, 1 in 171, but the PMET1 variant was present in about 1 of 18 people she analyzed. This mutation was incredibly common. We're very surprised that it was so common in these patients. And we know that this mutation causes loss of function of the Citrin protein. So we think this may have a profound role in the disease. However, in her analysis, she could not find any homozygotes for the PMET1 mutant. We suspect that perhaps the homozygotes are maybe not viable, and that's why they don't occur. But anyway, the PMET1 variant was very, very common, and we wonder, well, is there some reason for this? It seems unusually high just for random chance. Perhaps this variant, this PMET1 variant, may have some sort of protective role. And so we investigate protective role against maybe an infectious disease. So we investigated this in some more detail. And as I mentioned, there is an alternative star codon that would result in the production of a truncated Citrin protein that we looked at, the delta 134. There's actually an upstream translation start site that's out of frame. It would produce a 21 residue peptide. So we wanted to see if all three of these translation start sites were functional. And of course, the wild type protein, well, when we mutated the protein sites, they only had one of the potential translation start sites, and we can see, of course, the endogenous translation start site indicated by M1, of course, produced a lot of the protein. For the M2, we inserted a one base pair to make it back in frame so we could detect it. And the M2, the second translation start site, was active, not as active as the first, and the third, which would produce that truncated delta 1 to 34 Citrin, wasn't very efficient at all. But we did see quite a bit of expression from this second translation start site that would make that 21 residue peptide. So we did a blast search of that peptide, this 21 residue peptide, just to see what we could pull up. And one of the strongest hits we got was for a protein called CLAG9 from the Plasmodium falciparum, which is the parasite that causes malaria. We thought that was quite interesting, that this mutation is quite common in Thailand, which is an endemic area for malaria. And this 21 residue peptide has a high homology to a region of the CLAG9 protein. Another interesting thing we saw when we looked at this in more detail is that people have looked at the CLAG9 peptide as a potential vaccine source. Peptides made from CLAG9 are known to inhibit erythrocyte infection by the malaria parasites. So what we imagine might be happening is that cells that express the PMET1 Citrin may be expressing this M2 peptide. Perhaps this may limit the ability of the Plasmodium to infect cells. Now, one thing I didn't really mention clearly is Citrin, or AGC2, is expressed almost exclusively in the liver, so we think it would have to be having an effect in the liver stage parasites. So CLAG9 is known to be involved, so far they've tested erythrocyte infection, but the presence of CLAG9 or CLAG9 peptides inhibit erythrocyte infection by Plasmodium. We suspect that a similar thing may be happening in liver cells. Perhaps as liver cells are infected by the Plasmodium, they may rupture and release this M2 peptide, and that release of the M2 peptide may prevent infection of subsequent liver cells. So we don't think that the expression of this M2 peptide or the PMET1 variant would prevent malarial infection, but we think it may limit infection or may limit the severity of infection. And our collaborator, again, Ajahn Dwangreddy, is working together with the Faculty of Tropical Medicine to look at the phenotype of patients who have had malaria. So they have the malaria infection. They have samples from people who have had malaria, and they have indicated if their infection was mild, severe, or moderate. We want to see, is there any correlation between the PMET1 variant and the severity of malaria infection? And that work is ongoing. We don't have any results, but we think that this may well be protective against Plasmodium infection. We're also going to synthesize this peptide and do some in vitro infection assays and see, can we directly see a change in Plasmodium infection for both erythrocytes as well as hepatocytes. So just give you a quick summary of the first part, and don't worry, the second part's quite short. So the three novel Citrin variants we found, only the PMET1 is thought to be pathogenic. But we did show that we can characterize Citrin variants quite well and understand whether those variants are disease-causing or simply variants with no effect on the protein function. So again, we think this will help in the diagnosis of patients to determine whether or not they really have the citrin deficiency disorder. And again, the PMET1 variant is very common in the Thai population, and we think there may be some kind of protective effect of this 21 residues expressed from that second translation start site. So the second one, we were looking at genetic suppressors of the citrin deficiency to try to identify other targets or potential therapeutic targets. And the reason we are interested in this is, well, the Baker's yeast is a very, very great model system for looking at genetic suppressors. And suppressor analysis is useful because you can often identify unexpected interactions between biochemical pathways. The type of genetic suppressor we were doing was called a multi-copy suppressor. So we were overexpressing a collection of yeast genes into the AGC1 null to see if any of those genes would enhance the ability of the yeast to grow. So really the idea is that normally with the malate aspartate shuttle functioning, the cells can grow on acetate as a carbon source. In the Citrin mutant or the AGC1 deletion, the malate aspartate shuttle is blocked, no growth. But if you had another gene that was overexpressed, maybe it would activate a secondary pathway. So we were looking for any pathways that may be able to compensate for loss of the primary one. So again, we transformed in this yeast DNA library into our AGC null cells. All these cells could grow on glucose as a carbon source, but we selected only for those that could grow on acetate. As I mentioned, acetate was mimicking the carbohydrate toxicity seen in the citrin deficiency patients. So we looked for only mutants that could grow on acetate. And we think that any of these mutants may increase the activity of the malate aspartate shuttle or activate another compensating pathway. So we found about 92 potential suppressors after screening around 8,000 colonies. We haven't completed this screen. We need to screen around 50,000 colonies in order to complete the coverage of the genome. So we're only less than a fifth of the way through our genome screen. But we found about 92 potential suppressors. We narrowed that down to just a few, around 6. And one that we found that consistently showed the enhanced growth was this MCS21. So we sequenced this clone, and we found this clone contained 2 complete genes. One was PEX34, the other was CAR4. So PEX34 contains a peroxisomal integral membrane protein involved in regulation of peroxisome population. The CAR4 is a transcription factor that's involved in the response to pheromones, but it actually was the PEX34 gene that seemed to be involved in the suppression. And how PEX34 is thought to function, it works with several other proteins in sort of elongating the peroxisome neck, and increased expression of either PEX34 or another gene, PEX11, enhances this sort of constriction and division of the peroxisome. So we thought perhaps there's some connection between the overexpression of PEX34, peroxisome proliferation, and the suppression of the citrin deficiency. So we looked at the the peroxisome number in our cells that had overexpressed PEX34, or we also generated a plasma that would overexpress PEX11. And we could see what we predicted, that the peroxisome population was enhanced in the PEX34 as well as in PEX11. Though it's not identical, what we noticed in the PEX34 cells, instead of having several large mature peroxisomes, these cells had many, many small peroxisomes, sort of immature peroxisomes. The PEX34 had many more peroxisomes, but they were the sort of the large mature form. So we think that perhaps something about this peroxisomal proliferation is probably tied to the suppressor event, and we're currently trying to show if the peroxisome proliferation is sufficient for the suppressor to have its effect. One reason that the peroxisome seems to be a very, very interesting metabolic pathway that may be involved in the suppression is there's actually a lot of crosstalk between the mitochondria and the peroxisome. Metabolites such as the TCA cycle and galoxylate cycle are made both in the mito and the peroxisome. Fatty acid metabolism occurs in both the mitochondria and the peroxisome, but what we think may be most important for our question is actually the redox shuttles. There's the malate aspartate shuttle in the mitochondria. There's also a malate oxaloacetate shuttle or a glycerol 3-phosphate dihydroxyacetone phosphate shuttle. We think perhaps the peroxisome proliferation may increase the activity of these other redox shuttles, and maybe that increased activity of the peroxisomal redox shuttles is partially compensating for loss of the malate aspartate shuttle. All right, so that's where we come to the end of our story here. We think that PEX34 and also PEX11 may be high copy suppressors of the AGC1 cells. Should note that these suppressors are not especially great suppressors. They compensate to about 20% of wild type activity, but they may be enough to give us some clue about other pathways. And of course, we're continuing to screen the yeast genome to see if we can identify other suppressors. We think that these may be functioning through activation of alternative shuttles that will assist in the reoxidation of the NADH. And we're hoping to continue with these studies, and maybe we can identify some additional suppressors, or again, try to understand better how PEX34 or PEX11 are functioning as suppressors of the citrin deficiency disease. So of course I have to thank all the people who are doing this work. So most of the work was done by two graduate students in the molecular medicine program. It was Parath and Cholong Chai. And also I have to thank our collaborator from Rama Hospital, the Department of Pediatrics, Ajahn Gwanbridi, and of course all the other students in our lab who are helping with this work, as well as the Faculty of Science and the Faculty of Medicine, Rama Hospital, for their financial support. And I'd like to thank you for your attention, and if we have time, I could answer any questions.
Thank you.